Hi, welcome to the Bioinformatics Chat. This is the second part of a two-part conversation with Sergey Nurk and Sergey Koren about genome assembly. In the first part, which was in the previous episode, we talked a lot about genome assembly in general and about HiCanu. So if you haven't heard that one, that may be a good place to start. And in this episode, we'll dive deep into the world of HiFi sequencing and HiCanu. So now let's talk about HiCanu. And HiCanu is a version, a modification of HiCanu that is specifically tailored for HiFi reads. And so I think a good place to start is if one of you could give an introduction what HiFi reads are and how do they work.
Sure, I can start, and Sergey, you can fill in anything I miss. So I'm not sure how familiar listeners are with PacBio sequencing, but for any of their sequencing, you ligate these smart bell adapters to your molecule. You have your DNA that's double-stranded, and it has the ends, and you attach this little loop on both ends so that when you put it into the well, the ZMW that PacBio is actually using to observe the polymerase, as the polymerase starts from the open region there and starts reading along the molecule and separating it and synthesizing the complement strand. When it gets to the end, it just will go around the circle and start going again on the reverse complement version of the sequence. So essentially, as long as your polymerase lives, you will keep sequencing the same sequence first in the forward strand, then the reverse strand, then the forward strand, the reverse strand, forever until you turn off the instrument or your polymerase degrades. And so in typical PacBio sequencing, what they call CLR, these continuous long reads, you try to load very large fragments like 40 kb, 60 kb, and you start right away, you put them in to ZMW and you start reading almost immediately. You turn on the laser and try to observe the polymerase. And because you're starting with this DNA that has to be separated, right, because it's double-stranded, it takes the polymerase longer to go around the first time than it does subsequent passes. And during that time, it's getting damaged by the laser. And if there's also DNA damage or other issues it may just fall off the molecule, and so it'll stop reading. And so typically in the CLR, you'll read each molecule that you've loaded, one time or even less, right? You might not make it all the way around. What HiFi sequencing does is there's a couple of ideas. Is one, you select for shorter fragments rather than trying to get 60 kilobase fragments. You select a tight distribution of something like 20 kb. And rather than just turning on the laser right away, there's a time where you run the instrument blind, where you wait for the polymerase to go around the molecule once, which is the one that takes the longest time. And then you turn on the laser to observe it, and then you can see it's going to go much faster. So you will have a chance that while you're observing the polymerase and it's being degraded by the laser, it'll have a chance to go around that 20 kb molecule more than once. It'll go around, 5-10 times because you've waited for it to make sure that it can go around that first long time, right? And so these two ideas let you do a couple of things. So the first thing you do is that you kind of overload your ZMWs, so that because you know that some of the molecules will have DNA damage, have some other issues, the polymerase may fall off them. And so if you put two molecules into a ZMW, normally that's bad because you can't tell which one is being sequenced by the polymerase. But if you have a good chance that one of them won't work with the reaction, you don't have to worry about that as much, and so you expect that only one of them will be continuously extended. And so you do this, and since the polymerase with their engineering changes, their polymerases can survive for something like 200 kilobases of sequencing time, essentially. You can read the same data 10 times. It's the same 20 KB read 10 times. And so once you do that, you have the information that all these reads that you read 10 times came from the same place in the genome. So you can just pile them up on top of each other and call a new consensus and say, well, here's a much better version that represents that region of the genome because I know these all are from the same locus and my errors are pretty random, so I can get most of them out by just reading over and over again and seeing that they're different, right? And get a better accuracy read. And so that's essentially...
Or so we thought.
Yeah, well, modulo a couple of systematic issues, but for the most part, the reads are very high quality. And so essentially, you can think of it, it's analogous to if we're doing correction like I introduced in HiCanu, where we try to find the overlaps between all the raw reads, and we then call a new consensus for a given read using every overlap we found for it that we deem good enough to use. Except now we have a magic oracle we can ask and say, did these two reads really come from the same locus of the genome, which normally we don't know. We mix in different haplotypes, we mix in different copies of a repeat, just because our overlapping is not perfect. And you can think of the HiFi data as essentially a perfect oracle for that first correction step, where it will not ever mix haplotypes. It will not mix repeat instances. If you get a read from repeat instance A, it's not going to make a consensus of repeat instance A and B. And so that's very powerful for both genome assembly and for separating haplotypes and other kinds of applications where you care about, differences down to 1 in every 1,000, 10,000 bases.
Right. I also wanted to mention that the approach was there for a while, right, Serge? So the PacBio have been producing CCS reads for a while, but they used to be limited. Their length used to be limited by like 800 base pairs or 1 KB, which made them not very popular choice. Because people would rather get, like 7 KB noisy reads because they were more, they were, for most of the assembly, at least, they were more useful. But the key difference now is that these are the same high quality CCS reads, but now they are 20 KB long.
Right. So I think, So essentially, if anyone's familiar with IsoSeq, essentially IsoSeq was built on CCS reads. They were just on the order of a couple of KB long. And I think the keys that allowed this new 20 KB HiFi CCS reads to exist is that the polymerase has gotten more stable and better, so they can survive for 200,000 bases of life of reading. And B, that PacBio figured out these pre-extension, this running blind step, and good loading and library selection in a way that lets you get good throughput and yield from your CCS data, because it used to be that also you would get a very, very tiny fraction of reads that became these CCS reads. Right. So when you load your library, you would only have like 10% of your data be CCS reads or much lower. And that hurts your throughput. So you want to, so I think they've optimized those kind of conditions.
Okay. So basically this is a new protocol that gives you much more accurate reads. The main issue with long reads for a long time used to be that they have a relatively high error rate, and this allows to bring that error rate much further down. And you'd think that if you have better quality data, then the same algorithms that work for worse data would also work for better data, right? They don't get worse from working with better data. And so you could just continue using HiCanu on this new type of data. But I guess the trick here is that as our data becomes better, our expectations also grow, right? And so which features or which design decisions made in HiCanu made it not the best fit for HiFi reads.
Also, I think it is, HiCanu is a natural fit for the HiFi data. And the first paper that presented this HiFi data type, which was recently published, did use HiCanu along with all the other standard long-read assemblers that have been developed for noisy data like Falcon and WTDVG2. And, they all work reasonably well. The issue is that we're essentially trading one thing for another. So the long reads, while they were quite noisy on an individual level, when you got to the final consensus of the assembly, it was pretty good. And because they were so long, there were a lot of repeats that even though they were very similar, you could still resolve them because the reads were longer than them, right? So you can imagine that any repeat that's 50 kb long, if I have 60 kb reads, is trivial for me to resolve an assembly graph because I have unique anchors on both ends.
I actually don't think that we mentioned it before. So
that's true. Yeah.
that there are very long reads now. So HiFi are 20 kb long, which is very long, especially for somebody who comes from short read assembly background. But at the same time, there was this process of long reads getting longer. And Oxford nanopores, all these, people who are watching whales, which are essentially reads that are longer than 500 kilobases, occasionally get them, and long read, ultra long read sequencing protocols, which give you N50 of your reads, well beyond 100K, I believe. And even for PacBio reads, right, which many people prefer due to more random sequencing errors and higher resulting consensus, they were also getting longer and longer, and now you can get CLR reads which are up to, 70 kb long, I believe. So compared to that, those lengths, 20 kb is somehow already seems pretty short.
Right. And so that brings us back to this idea that, the assemblers were primarily designed to address the issue of, well, how do I find similarity between these reads that are 10%, 12% error, right? And then we got resolution of repeats largely because we had reads that were longer than these repeats. And, in the case of a recent project where we're also part of this telomere-to-telomere consortium where we're trying to finish the human genome. And as part of this project, we obtained these ultra-long nanopore reads. We actually had something like 40 or 50-fold coverage of reads greater than 100 kb, which drastically simplifies assembly. And so when you give these assemblers the 20 kb reads that are very accurate, they're still very, very short by comparison. And so the question is then, well, how much accuracy do you need to gain to compensate for the read length? So essentially you can think of it as a question of, if I have a 200 kb read that's 10% erroneous, what length, 99.9% accuracy read do I need to match its assembly quality in terms of repeat resolution, right, on some genome?
Right, and the answer would be different for different genomes.
Yes.
Right? So, so what we were mostly looking at at human genome, actually weird human genome, which is CHM13, and it is effectively haploid human cell line. That the T2T project that Sergey mentioned, which tries to fully assemble all human chromosomes. This project is focused on this genome, so that was our model for now. But I think you formulated it perfectly. That indeed we do not expect the tools to start perform worse on these datasets compared to the CLR dataset, for example, or Oxford nanopore dataset with the same read length, right? But since we now have this technology and the reads became much more accurate, but the assemblies did not become much better. So the consensus became higher, the continuity became somewhat higher, but without any additional development, it wasn't, a revolution. And since HiFi now needed to compete with ultra-long read technologies, also from PacBio itself, so like much longer CLR reads, there was no strong opinion on whether one actually should generate HiFi reads to get or whether it should be a technology of choice for genome assembly. So it could be useful for, haplotype analysis still, but so that's where we started. I guess this is where Serge started experimenting with HiFi reads, because these initial experiments predate me joining the lab.
Yeah, and I'll go even further to say that, I personally was quite skeptical of the HiFi data too, given, the large size discrepancy, and that the reads were not, even though their accuracy was quite high, they were not perfect coming off the sequencer. And we could see that they did still have mistakes, kind of in the places where you expect from the long reads. So one place where they have a particularly high error rate is in the homopolymer stretches. So if you have 8 A's in your genome, you'll have a bunch of reads that say there's 8 A's and a bunch that say there's 9 A's and a bunch that say there's 7 and some that say there's 6 and some that say there's 10. And so unless you did something to the reads within the assembly we didn't think you could exceed the quality of these ultra long assemblies that are coming out of the ultra long nanopore data that we had.
Actually, I remember that, and it's funny how it changed overnight. So, right. Yeah, but we will get to this, I guess. But yeah, so Serge was indeed super skeptical and was pushing towards us using ultra long nanopore reads for the foreseeable future. But I was very interested in HiFi reads. Interestingly, mostly not because I thought that it's a good choice for vertebrate assemblies, but I was very interested in their application to metagenomic data. And so, since I had this interest and I thought that they should be a very good fit, I was very interested in digging deeper into into how far we can go in their assembly. So. This is what mostly fueled my interest, not the perspective of, getting almost perfect assembly of human chromosomes. Although, right now we are at a very different point where we now can assemble human chromosomes very well, and I'm still struggling with assembling, with getting good assembly for not so complex metagenomic community. So it's kind of reversed.
Yeah, so the seeds of HiCanu were, there was an engineer at PacBio, Rob Gothe, who is a co-author on the HiCanu paper, had suggested that I experiment with homopolymer compression of the reads, since we knew that these homopolymer stretches that I described before were one of their main error modes. And so those were kind of the early experiments with the proto HiCanu, where we added the ability to compress the reads within Canu, and then we would operate in this compressed space. And one thing I had seen before Sergey joined and started working on this project is that we indeed could do much better in terms of resolving repeats, very similar repeats like segmental duplications in the human genome. But we had very bad continuity. So we were getting the hard parts, but we were getting the easy parts wrong, or we were, fragmenting the easy parts. And so that kind of was the start of Sergey's involvement, I think, where he really took off and started looking in detail at this data and trying to understand and comparing, why did we make a mistake in this easy part? Why did we break this easy part that should have been, which was previously assembled correctly? And it's not repetitive. And so he found other issues in the data that I can let him describe, the process he went through and the kind of the changes he made to Canu that cumulatively decrease the error of these reads. So starting initially when you get them off the sequencer, when we map them to what we think is a good truth set for the CHM13 genome, the chromosome X that we resolved semi-manually, that none of the reads map perfectly. So all the reads have some mistake. And then after you go through all the processes of correction that HiCanu uses, 97% or more of them have no mistakes. And so that's a dramatic improvement, right? You go from every read has a mistake to almost no reads have a mistake, which obviously is very important for assembly, because now we can say, well, I have a 20 kb read that I'm pretty confident. If I see one difference between two reads, that's probably not coming from the same locus of the genome. So now if my repeat has only one difference every 10,000 bases, then I can be confident and I can actually separate those repeats, which is what's enabling these extremely high quality assemblies of human chromosomes and other similar kind of repeat structures in other genomes that have these large, slightly diverged, but very large repeats.
Right. So to make this idea a little bit more explicit, let me know if my understanding is correct, but the repeats cause problems for assembly, because in the assembly graph you have two edges, let's say, entering a node that corresponds to a repeat, or a read from a repeat, and then two edges leave that node, and you don't know how to pair them. You know that those are two different regions that have this repeat sequence, but you don't know whether the upper edge should be paired with the upper leaving edge, and lower entering edge should be paired with the lower leaving edge, or vice versa. And so when you have very long reads longer than the repeat, then those reads cover the last part of the entering edge and the initial part of the leaving edge, and that allows you to resolve the repeats, meaning pair those entering and leaving edges. And that's one strategy that you can use when you have these ultra long reads. But with HiFi reads, the compromise is that they're shorter, but they're more precise. And so you use the different strategy, which is to find these tiny differences, because the repeats are not perfect. And so if you can spot these differences, then you can also resolve these repeats, because they are essentially no longer repeats. They're different sequences. And so in the paper, you have an interesting set of numbers for how divergent the repeats ought to be in order for different technologies to resolve them, right?
Yeah, I will let Serge comment on this modeling part, because he likes it. It's not as simple, unfortunately, but I wanted to comment first on the repeats part. So you are exactly correct, but modulo the fact that you are thinking about it very much in terms of, repeat graph of the genome. So when you see where repeat is, actually the way that repeat represents itself in the graph depends on what graph module you use. And so in overlap graphs, the repeats that are shorter than read length will look differently, actually. And we'll have this, what's called often referred to as hourglass motif. But it doesn't matter. The idea is the same, that during assembly, you find overlaps between the reads. And if you have, for your read, you have several different overlaps which disagree with each other, right? So it overlaps with two reads that cannot follow each other in the genome. You often cannot choose essentially which way to go. So which one of the reads should extend your read. So all assembly, I think, can be thought of as, gluing reads based on their overlaps. That's essentially what's going on, independent of which model you use. And so you just don't know where to go. And then with the long reads, again, depending on how you represent your graphs and so on and so forth, but the idea is that everything becomes simpler if you have a read which anchors to unique parts of the genome. So the parts which are not repetitive, for which you do not have homologous regions of high similarity in the genome, right? So whenever your read spans the complex region of the genome, you're good. And with high, there is, so the way you started describing how to use differences between the repeats, actually there are tools which do it exactly like that. So when they actually try to spot places where the reads differ from each other, so they, for example, first get the initial representation of the repetitive region, like consensus representation, right? And then try to align the reads and see where the differences are, and then to understand, so here probably there are 3 copies, and here are the particular differences which are specific to each copy, and then, okay, we now can resolve this part. This is what Fly does with the repeat resolution. We have a different approach. So in our approach, essentially, what is a repeat? Repeat within the assembly is everything for which you identify the overlap. For example, imagine you are only looking at the perfect overlaps without any differences. Then if the repeat copies have differences every, like, for example, 100 base pairs, you just won't see it at all. And it won't confuse your assembly because the reads from different copies of the repeats won't overlap at this, won't overlap in this case. Right? And so, this is, I think Serge actually missed this part. So, the reason why, the assembly stopped being continuous is not because the data was bad. It's because he was super ambitious. And he realized that, so he knew all too well that depending on this threshold that you use for detecting overlaps, right, you implicitly immediately resolve many overlaps. So the higher the threshold that you can use on your overlap identity, the better will be your results. And so he tried to use very, very stringent identity, which seemed for me crazy at the moment. So I didn't think that we would be able to do that. That's why his assemblies were more fragmented. So the previously published HiCanu assemblies and the other assemblies that we used as baseline, they were actually well. But we wanted to do better, right? So we wanted to resolve these hard segmental duplications. And so what Serge started seeing is that when he increases the threshold he uses to detect overlaps, the number of resolved segmental duplications goes up, but the overall continuity goes down. So yeah, that's when I joined the game. Yeah. Anyway, I think we need now to discuss the second part of your question, right?
Yeah, so this repeat modeling, so it's specific to the human genome. And what we did, this was actually motivated by our work on the paper that had some of the first ultra-long nanopore data, where we were interested in the question of, well, how much ultra-long nanopore data should we be generating? And so we wanted to try to answer this somehow without actually generating the data, because at the time it was pretty intensive to generate. And so I used the current human reference, this JRCH38, which has a lot of different annotation information and tracks that are available through the UCSC genome browser that people have contributed based on various analysis that give a good idea of what the repeat structure in the human genome look like. And so what that model does is it's really simple. It takes all that information and collates it together. And so then it has a catalog that says, okay, at this position in chromosome 2, there's a repeat that's 98% identical to something else in the genome. At this position in chromosome 5, there's a repeat that is 50,000 bases long, and we don't know what identity it is because it's a gap in the reference, let's say, and so we assume it's 100% identity. And so then what you can do then is say, okay, let's assume that we can resolve any repeat that's has more than 2% differences. We're just going to say, okay, ignore that. That's not a repeat as far as we're concerned. And then we look at the other repeats and say, okay, if we had a 50 kb read, how many of these repeats would they span, right? If we had a read that was 20 kb, how many of these repeats would it span? And so then you just plot that information for various read lengths, and you get this kind of line that tells you what you expect to see from your assembly. In order to put an actual assembly onto this plot, what you have to do is look at the actual read distribution. And because the reads aren't all the same length, you do a little probabilistic that you have a good probability chance of getting a read at least this long, right? So if you have 100 kb reads, that doesn't mean all your reads are 100 kb. So you do have to do a little bit of statistics just to say, okay, if I have 5x of 100 kb reads, I'm not going to cover every possible repeat, but I'll cover a lot of them with this length read. And so typically from what we've seen empirically, when you take real assemblies and put them on this plot, they seem to follow something like they can resolve repeats that are more than about 2% or so diverged, maybe a little less than that, somewhere in that range, from long nanopore data or from the PacBio CLR data. And when you look at the when you take these assemblers that were designed for the long read data and you apply them to the HiFi data, they seem to follow somewhere between below 1%, something like 0.5% of divergence of repeat is enough to resolve. And this makes sense because these reads are about Q20. And so, if you think that the overlap is about twice the error of the individual read quality, typically the assemblers run at somewhere like 1% tolerance for differences, 0.5%. So any repeat that's more similar than 0.5% is going to show up as a branch in your graph, is going to show up as something that confuses your graph, where you get these reads, like Sergey said, where you have incompatible reads and you have no way to figure out which one is which. And so, based on that same modeling, we saw that if we could improve the read quality, you should expect much, much better assemblies, because there are a lot of near identical but not identical repeats in the human genome, according to our model. And actually, there's even more of them than the model says, because as I mentioned when I was introducing the model, when you have a 50 kilobase gap in the reference, my model just says, well, that must be 100% identity. But obviously in real humans, that's not true. We had some earlier work in the lab led by our postdoc, Alex Stilthy, and the collaboration with NCI that looked at the rDNA regions of the human genome, which are these large tandem repeat arrays of 50 kb. And we saw that even these regions that are under high pressure, right, because these are highly functional genes, they still had variability and variation that were present at some relatively regular intervals, perhaps more than people would have expected if you asked them. And so because there is probably more variation than represented in our model, our assembly, at some point, our model becomes kind of useless when your assembly is resolving really, really similar repeats, because the model always says you can't resolve that repeat because I have no information on it, which is not always the case in a real genome.
Right, but it's not that like we've been checking with this model, to set up the thresholds
No.
while we were trying to look at what's going on.
No, it's more of a motivation of like, if we could only have reads that were this good, we could do better. We should be able to do better, right?
Right. Yeah. And so we started looking at why we're not doing better. And I started at looking at the points where our context broke, essentially when we try to increase the identity, right? And I started seeing, first of all, I started seeing some errors in the reads, which was expected. the not so expected part was that HiCanu should have been able to deal with them because it had this special module called overlap error adjustment, which doesn't matter. But essentially, HiCanu, as I said, HiCanu has everything there, right? Everything is somewhere in HiCanu. So it also had some procedure for read correction. Yeah, I actually was considering other strategies as well, like trying to detect the gaps, so the reads which we couldn't extend, and then lower the identity thresholds at those regions, so that we will connect it to somewhere, and it's some procedure, some kind of gap closing procedure essentially. But then I started, before implementing anything of that sort, I decided to understand why the module wasn't working well.
So the read correction module, is that the thing that we discussed in the first part of this discussion, where there are overlaps and they correct the
right? So it's similar, but it's different. So it's a different module, and the difference is that in that initial correction, we always take the consensus of the overlaps which we got. So we do not try to actually analyze the alignment itself. So we get the reads which align there, we take consensus, and we spew out the new version of the read. So this module was trying to do something less disruptive, and it tried to look at the pileups of the reads and only correct the base if all of the reads at this position were agreeing at the base which we should use there, right? Instead of the one which is in our read. So instead of just taking consensus, we were not introducing the changes if the current state was supported by some reads. Well, not by single, but by two reads. It doesn't matter. But in the sense, it's very similar. But I think, yeah, right. So the important part, the initial correction we disabled immediately, because the initial correction would kill our efforts to disconnect the repeats of high identity, right? To resolve them, because we would get some reads with weird consensus, which would be some mixture, some chimera between different similar copies of this repeat. So that initial part we disabled first, I would guess, right, Serge?
Yep. Yeah. Essentially, the homopolymer compression is what we do instead.
Right. So we thought that homopolymer compression would be enough. It was not.
Let's define that. That is a very simple concept, but let's define it formally.
Oh, homopolymer compression? Right. So it's also called. So similar one is called run length encoding, and it's more actually, I think, better notion. But we use homopolymer compression because we do not log. So if you have a stretch of the same letters, run length encoding says that, okay, so how many of these letters you now have. So instead of having letter A repeated 10 times, you just write down A10. And in homopolymer compression, you just write A. So we are forgetting how many copies of A were there in the first place. And we do this transformation on all the reads and actually then perform the assembly in this homopolymer compressed space. I think very similar trick is used in Shasta assembler. So I mean, we are not the first ones to do that, and it works. So because both PacBio and Oxford Nanopore, as Serge mentioned before, this is the primary mode of errors to add insertions or deletions depending on the technology, to undercall or overcall the number of homopolymer bases.
Yeah, and so this procedure, if you have two reads that have, they differ in the number of times some letter is replicated, this procedure takes these different reads and make them similar, although they now have very little in common with the true sequence, but at least between themselves they are now identical.
Right. And also, I mean, it's important to understand also that we are actually masking the real differences, home polymer differences as well. So if we have two repeat instances that differ only by the number of times the letter A is repeated there, we won't see this difference anymore. Luckily, usually repeat like segmental duplications have different types of differences there, right? And actually the primary mode would be not home polymer error, but would be substitution of one letter against to a different letter, right? So that's why we are along with ignoring most of errors, it allows you to still see most of the real differences. So I started looking at that, and then we realized that if we fix some of the remaining errors, we would have a chance to essentially not make a gap at this position. And so I suggested doing read correction, and then Serge told me that we already should have read correction. And then I started looking at why it wasn't working. And apparently this module that hasn't been very useful for the results on PacBio and Oxford Nanopore reads, it was broken in at least, I think, 3 or 4 places. And it's a very cool example of how, if you have a low selection on some part of your code, so essentially if it's not essential, it accumulates bugs at a drastic speed. And the final bug that was there was particularly hilarious because it essentially disabled the entire module, and nobody knew nobody knew that it happened. So but
I think it was not working properly from the start of Canu, right? I think it got broken when it got moved from Solera Assembler into Canu.
Well, yeah, there was one bug. Then you introduced another bug while trying to fix some segfault, right, which essentially disabled it in like half of the cases. But then its results, right? But its results were entirely ignored. So, and then came the cool thing, and I think it's important to thank Serge for that. Serge has this cool property. He does a lot of experiments. So, I would obsess for resolving some particular cases, and to trying to make this part perfect and to dig deep into investigations. And so I was investigating and I was fixing. So I made one fix, I made the second fix, I made the third fix. And then while I was fixing that, Serge was, essentially running the assemblies every day, just on the version that was most up to date at this point. And then, after one of the fixes, it just worked. So essentially, we wake up in the morning to see that we now have one of the best assemblies of this genome that were ever, made, but without any additional efforts on our part.
That sounds like a typical Hollywood scene, from a movie, when a hacker just wakes up and sees that all the code has been fixed.
Right. So I wasn't expecting that, because at this point I was, fixing bugs in Canu for a month and a half or something, and they were I thought that it would be a never-ending process, but then it clicked. So it was a very cool moment. Immediately Serge said that okay, now we don't care about ultra long nanopore reads. Let's focus on this stuff. and so then we did some additional work. So we, for example, identified some recurrent errors in the HiFi reads, which were not described before, and essentially we realized that not only homopolymer runs cannot be trusted, right? But also the number of copies of simple pattern in micro,
like a dinucleotide, like AT or something like that.
Yeah. In this simple repeat arrays.
Microsatellites?
Yeah, microsatellites. Thanks. Sorry, brain freeze. So they also couldn't be trusted. So we, instead of digging too deep into this, we just decided to ignore the differences in those regions while we are comparing the reads to each other. Right, so when we are trying to understand how good overlap is, well, that obviously ignored some more real differences between repeat instances, and I think this part can be improved. But yeah, at this point we started already getting very good assemblies, and then we had to introduce some changes within Bogart because. Essentially, it was lacking the procedures which are, standard for most other assemblers.
Let's recap briefly what Bogart is. So this is the way you choose overlaps?
No. It's this module for essentially building contigs from the overlaps. The one that uses best overlap graph. It's Bogart because it's Bog based on best overlap graphs.
Right.
And it's art because it's heuristics that no one knows. Art doesn't actually stand for, as far as I know, it doesn't stand for anything. It was originally Bog, and then when they adapted it to Lumina data, it got renamed to Bogart. There was a developer of Stellar Assembler named Art Delcher, but I don't think it's named after him because he was not actually actively working on this code. So I've never heard an explanation for why it's called Art.
I like your explanation.
Yeah.
It's true. Well, now it's not true already because since we submitted the paper, actually most of the time we spent afterwards was on trying to figure out what Bogart is actually doing and, making it more reliable and
robust.
Yeah, for example, it was skipping a lot of, tandem repeats, just putting essentially a single copy and then going further without any evidence for doing that and things like that.
But I think one of the major changes we had to make to it for the HiCanu paper is handling heterozygosity better. So this human that we were working with first, the CHM13 cell line, it's like Sergey mentioned, it's effectively haploid. It has 2 copies of every chromosome, but they are the same everywhere, or for the most part, with a couple of exceptions. And so when you assemble a diploid genome, you get, in this best overlap graph, you get a long path. Your first greedy path is going to represent some pseudo haplotype where you randomly walked through and you switched between the different haplotypes. And then you get these shorter stretches, which are the kind of bubbles in a traditional graph. But because you have a best overlap graph, you don't actually see them as bubbles during the construction because you don't look at all those overlaps. And because Bogart had this step where it went back and looked for branches, it couldn't tell the difference between a branch that was caused by a repeat, a real issue, or if it was a branch caused by a bubble. And so it had the effect that on very heterozygous genomes, it would overfragment the assembly. And this was a problem on genomes that were diverged, from the PacBio long-read data or from the Oxford Nanopore data. If they were more diverged than a couple of percent, because that was, essentially everything below that we just smashed and we didn't see that as a difference in haplotypes. We just made a consensus of both haplotypes and didn't care about bubbles, right? But once you got diverged enough, it would HiCanu tended to fragment those kind of assemblies more than it should have.
Right, but this is very HiCanu specific issue. That's important to understand. So in like other models, or it would be so that all this would end at
bubble detection.
At bubble detection, or super bubble detection. So But somehow Voger did not have this part at all. So we had to introduce it there, and it's a mess just because of how it's. I mean, Voger is old, and some of the later developed concepts they end up being tricky to introduce there. That's one of the reasons why we want to essentially, at some point go to some other way of producing the contigs. But importantly, so I think takeaway message here is that, if you don't allow, essentially, if you look at 100% identical sequences when you are searching overlaps, all the heterozygous differences, which you could ignore before, now you will see them. And this is also, so this is a pain if you want more continuous assembly that of a what's called pseudo haplotype, where you are okay with switching between haplotypes and one consensus. But it's also important because it allows you for diploid genomes to produce very long stretches of individual haplotypes, right? Just implicitly. So without any, special modules for haplotype resolution, just because you only allow for when you're searching for overlaps, you do not allow for any differences. Only reads from the same haplotype will glue with each other. And this is one part of the analysis that we showed that, essentially we are, for example, for MHC complex, we correctly reconstruct all the alleles of all the genes, which people usually care about when they look at this complex, but we cannot, just based on the HiFi data we have, because of its limited length, and because there are some regions of very, very high, low heterozygosity, right? So high similarity between haplotypes, we cannot, without additional information, it seems impossible to recover the fully resolved haplotype for this region, but we are reconstructing alleles of all the genes perfectly, all the copies of all the genes, but we don't know how to relate many of them. So we know how to relate some of them, obviously, but we cannot go through the entire MHC. So for this, we would need to integrate some additional information, like HiC or strength-seq or something else.
Trios,
yeah. Oh, yeah, right. With trios, we are getting perfectly assembled MHCs, I believe.
But without this additional information, those are just different contigs in your output?
There's one large contig that has a pseudohaplotype. So, there's these 6 canonical genes that we're using that are commonly the ones people type. And so they're spaced so that there's about a megabase of space between some of the genes. And so these genes, let's say there's A, B, C, right, genes. And so you will have A from the maternal haplotype, but you'll have B and C from the paternal haplotype in the long contig. But you'll have other shorter contigs that'll have the corresponding allele that you didn't have. So if you had the paternal A, you'll have a short contig that has the maternal A. And if you had the maternal BC, in that large contig, we have a short contig that has the paternal BC. But you will still have one large contig that's going through the MHC. It's just that, as Sergey is saying, we don't know how to tie the maternal A to the maternal BC because there's a stretch of homozygosity between them that we can't tease apart. And so we just will randomly connect the paternal A with the maternal BC.
Right, but if PacBio comes up with a way, for example, to increase the read length twice, maybe it won't be a problem anymore. Every update to the technology here could become a game changer because we are producing quite long pieces of these haplotypes, right? Perfectly phased.
Right, they're almost the megabase in length on a human genome, which is pretty good given the 20 kilobase reads. So, most of if you're looking at a gene, because these stretches are a megabase long that are phased, the gene is probably going to be all in one phase. So if you're looking at gene A, it's going to be all maternal or all paternal. It's just the correspondence between gene A and gene B a megabase away is not necessarily the same.
Yeah, but this is quite specific to how HiCanu output is organized. So, for example, in the other project by Heng Li called HiFi ASM, which he's actively working on, if I remember correctly, they produce like different kinds of outputs, and there you can, depending on what you want to do with those, you can choose between different kinds of outputs, and either see, this long pseudo-haplotype contigs, or really get, this graph with long bubbles representing alternative haplotypes, essentially. So, I mean, it's all work in progress, but it seems to be the takeaway is HiFi seems to be a very useful type of data to do analysis of haplotypes. And speaking of additional data types, I don't think we've mentioned that here that both recent works, which were trying to generate chromosome scale, haplotype resolved assemblies, were using HiFi along with some other type of data which would produce this, long range information. In one case it was HiC, and in another it was StrandSeq. But as a basis for their work, both of them were using PacBio HiFi.
Right, and part of the motivation for the HiFi, and something we haven't talked at all about, is that the HiFi is actually quite useful for variant calling as well. So both of those approaches that Sergey mentioned, they make a collapsed assembly first, and then they map all the reads, call variants, and call heterozygous variants, and then try to identify those variants within the reads, and then separate the reads into bins. And so there were similar approaches that were used before. Our postdoc Arang Ri was on this Korean human genome paper where they used combinations of technologies like 10x to call variants, and then they try to phase the PacBio reads using those variants. The nice thing about the HiC data is that you can get both from one data type. So you don't need to have Illumina to call your SNPs and then PacBio to do the assembly. You just do everything from the HiFi data, which is quite nice. And I think one of the most exciting things for me that was in the HiCanu paper that was actually a very late addition was we as we mentioned before, we're working on this T2T project. And so we have finished chromosome X, including the centromere. So it's the full chromosome. For the first time, we have a centromere assembled, and it's telomere to telomere. So there's no gaps. And we want to finish all the other chromosomes, obviously. And so there's ongoing work in the consortium to automate these assemblies. So there's work from the FLY team from Pavel Pevsner's group working on CentralFly and Mosaic Fly that are trying to improve the resolution of segmental duplications as well as centromeres automatically. And we were comparing some of our results, and I noticed that actually a centromere was assembled in the HiCanu output, which was very exciting, but I was very skeptical because, as Sergey mentioned, we knew there are shortcomings in Bogart. And so my first assumption when I see something like an assembled centromere out of an automated Canu assembly is that it's probably wrong, without good evidence that that's not the case.
We had good support for this assumption, right?
Yes, because
from the chromosome X, which was resolved but wasn't there
in the nanopore data. Yeah, in the nanopore assembly we had done with Canu of this CHM13 cell line, we had resolved quote unquote chromosome X, except it was just collapsed together. So we had a single contig that should have been split, but was not. And instead of having over 2,000 copies of this higher order repeat that comprises the centromere, there was like 500 or something, or 600. So it was obviously incorrectly collapsed. But our collaborator, Karen Miga, who is the co-founder of the T2T project, started looking at some of our centromeres and actually found that they matched her manual reconstruction. So she was doing these reconstructions where she identified all the variants in the centromere herself, and then made a tiling path of the reads that had these variants, that explained all the variants, that gave her a draft centromere with all the variants represented, but at a low sequence quality because it's just the tiling path of a single read. So it's the quality of one read, right? Using these ultra long nanopore reads. And so she was using that as kind of a benchmark, a baseline. And when she compared our assemblies to some of those, it turned out that our assembly was actually quite good and correct. And not only correct in one centromere, but correct in several centromeres. So we ended up in the HiCanu paper describing 9 of these candidates. Now, obviously, not all of them are going to be perfect. There are still going to be some mistakes in them that will need to be fixed, that will be fixed through the changes that Sergey mentioned that we're improving. Bogart, some of those changes fix mistakes we were making. And other improvements to the assembly, improvements to the data. But it's extremely exciting that, about a year ago, we were working on the T2T project and we barely had one chromosome with a centromere after lots of manual effort. And now we had 9 candidates, which, like I said, even if they're not perfect, it's much easier to at least have an idea of what the centromere looks like, what we're going after, what we need to fix when we have it for, almost half the chromosomes in that genome, than to just be going in blind.
Yeah, and actually there will be, we hope to get an update to this work pretty soon, where with, alternative and hopefully more robust method of generating contigs and assembly graphs, it seems that we should be able to get another T2T assemblies for another 5 or so chromosomes. So stay tuned.
So anything else you guys want to discuss about HiCanu?
So this is, I wanted to reiterate this important part that nowadays, if somebody wants to work with HiFi reads, it is, so most of the tricks and changes that we made to make it work can be reused by other people in their own pipelines, for example. So most of the changes that we discussed with you, they relate to how we correct the reads, how we score the overlaps, right? So actually it's possible to get these overlaps from HiCanu and do whatever you want with them. So I think it's important that if anybody wants to work, to experiment with HiFi reads, or to improve on our analysis, there is no need to be, professional Canu developer. There is no need to rely on all this code base. There are places where you can just grab intermediate results and try to do whatever you want with them. So yeah, if somebody is interested in doing some of this work, just contact me or Serge, and we will do our best to try to help you, get the intermediate output that you need for your experiments. I think would be my message.
I think that's a good point, and it's kind of cool that unlike a lot of the when you're working with the very noisy data, it's much harder to just grab the overlaps and go. It's much easier with HiFi data because they're so close. There's so few errors. It's much easier to look at places by hand and know what you're looking at than if you just grab the random stretch of back by long reads and try to look and make sense of what they're trying to tell you there. So I think it's a low barrier to entry to play with the HiFi data. And it's a very exciting data type. And
and it's super, it's very, fulfilling and very pleasant. For example, assembly graph from the entire human assembly, you can just look at it and try to see which regions are still hard to resolve and which are trivial, which would be impossible for, most of the other data types. You would just, get with this very complicated. So the whole situation will be just too complicated for you, to visualize or to, make sense of. And, apparently when you work with, high 100%, threshold for identity or for overlap search, everything becomes much, much simpler.
Yeah. I guess one other thing that's worth mentioning is one other thing we did discover that we describe in the paper is that at least currently there seems to be a bias where regions that are either large G repeats or GAJ repeats, they cause coverage dropouts. So there's some regions we found, like on chromosome 8 in our assembly that we looked at, and then several on chromosome X, where despite the genome being on average covered to about 30x, those regions were only covered by 2 reads or something like this, which you can imagine causes some difficulty for our assembly. And so this is an issue.
It's definitely non-random effect, because we saw the gaps. So we saw depletion of coverage at the same spots in other libraries as well.
So So there were 3 different size selections, different libraries of HiFi data sequenced at different times on the instruments, and they all have the exact same pattern. And so this is something we've communicated back to PacBio that they're looking at improving. So one of the reasons I bring it up is this is not a very common motif in the human genome. I mean, it occurs, but it's not, every other base or something like that. But there are certainly some genomes, it seems like fish in particular, have more of this type of repeat. And so if your genome has a lot of these repeats, the HiFi data may not at present work as well for you unless you somehow deal with these areas after the fact. So, if you assemble them with HiCanu, you'll probably have a bunch of gaps there. And you could then try to scaffold with the HiFi data or other data that doesn't have that issue. But that's just the caveat that, at present, there is this limitation of the technology that will limit its application to some genomes which have a lot of this motif present. And obviously, PacBio is working on trying to fix it because they don't want to have this limitation. But they were actively working on it the last time I had a chance to talk with them, but obviously they've been kind of shut down by
coronavirus.
That's right. Yeah. So, since California is pretty much shut down, they're not able to work on this as much as they'd like, obviously.
Yeah, as I mentioned, I'm also very excited about potential applications to metagenomic data. And I think it will be very interesting. And this data type should be able to help with, analysis of intraspecies diversity, especially in more complex communities. But it's all very early days. So the long read metagenomics is in quite, not very mature shape, and HiFi applications have been very, very few of them, and nobody actually knows what we will discover in the next half a year. So I'm very excited about that. I think it has potential to be, transformative for that area of research as well.
Guys, it's been an amazing conversation. I think these 2 episodes are a must listen for everyone who's interested in genome assembly, either as a developer or as a user. And thank you so much for being on the podcast.
Thanks. It was fun.
Thank you very much, Roman. You're an amazing host.
Yeah. Hopefully we didn't scare everyone away from using Canu now that everyone knows all the horrible bugs.